Hi, I'm Sophia, and welcome back to Deep Sea Mysteries. Today we are going to be diving into a paper discussing the genomics of giant isopods. I love these little guys. They are so cute. Gotta be one of my favorite deep sea invertebrates. When you think of isopods, most people think of roly-polies or pill bugs, Armadillium vulgare, a terrestrial species of isopod. There are actually a ton of isopods everywhere. They are the largest group of crustaceans, with over 10,000 species, and can be found on land, in intertidal zones, and in the depths of the ocean. At depths, we find the star of today's topic, the giant isopod genus Bathynomus. They are found at muddy bottoms at depths from 170 to 2,140 meters. The giant isopod is unique in its digestive system. It has a stomach that can expand to 2/3 of its body when filled, and fats that store organic reserves. They also can fast for over 5 years. As we've previously discussed, the deep sea is a hostile environment that isn't livable for many organisms. There's no sunlight, extremely high pressures, and low temperatures, not to mention the lack of food sources. Talk about a tough life. So, why do organisms even live here? How are they able to survive? That's what the researchers of the paper decided to find out. How did isopods evolve and adapt to the deep sea environment? These giant isopods, Bathynomus gemesi, were collected at 898 meters near Hainan Island in the northern South China Sea. The red pin is the exact coordinates given in the methods. Honestly, I'm not trying to get very specific here with the methods. Most of it explains DNA extraction, genome sequencing, etc., etc. I'll try my best to sum up the important bits. To sequence the genome of B. gemesi, molecule had to be extracted from the specimens. They used Illumina and PacBio sequencing to sequence the genome. Both of these help researchers visualize a genome that isn't perfect. They use a Falcon pipeline to assemble the genome and polish it using Quiver. Lastly, a benchmarking universal single copy ortholog, or BUSCO, is used to determine the completeness of the genome. To estimate the genome size before the complete assemblage, K-mer analysis is used. The researchers also did specific analysis with RNA transcriptome sequencing, repetitive sequence annotation, protein coding gene annotation, gene family analysis, phylogenetic analysis, and more. Bathynomus gemesi has a genome that is 5.89 gigabases long. That's huge. To put that into context, so far the human genome is 3.1 gigabases. The blue whale's is about 2.4 gigabases. Compared to our friend the pillbug, this giant isopod's genome is 3.4 times bigger. From K-mer analysis, they found that the genome of B. gemesi is about 89.7% repetitive, and from that, 98.7% were transposable elements, or DNA sequences that can move locations in the genome. This is the highest content of TEs found amongst crustacean genomes. These TEs were found to have a divergence rate of less than 20%, meaning they are pretty young. This shows the proliferation of TEs should have happened in recent history. The adaptation to the deep sea would have caused stress, which can lead to epigenetic changes, including this proliferation and a large genome. The proliferation of TEs can promote genome plasticity, which allows the genome to rapidly adapt to an environment. A phylogenetic tree was created from the genomes, which showed that isopods diverged from their last common ancestry 366 million years ago. B. gemesi and A. vulgare diverged 245 million years ago. This tree was supported by the fossil record. Many gene families in the thyroid hormone, insulin, and hipposignaling pathways in B. gemesi were expanded. An enriched incomplete thyroid hormone signaling pathway leads to an increase in growth and metabolism, which is in contrast to most crustaceans, including A. vulgare. In particular, this pathway influences growth through the alteration of the secretion of growth hormone. An increase in growth hormone can lead to linear growth and therefore giganticism. The insulin signaling pathway genes were identified to be tightly associated with body size evolution. Two specific genes stand out in this pathway, ISL and mTOR. ISL is involved in insulin secretion, metabolism, and mediates glycolysis. mTOR is the core gene of the mTOR signaling pathway, which plays an important part in growth regulation. It controls cell growth and metabolism in response to nutrients, growth factors, and cellular energy. Significantly more mTOR genes were found in B. gemesi than in other crustaceans. The hippo signaling pathway is an important player in controlling organ size, possibly explaining the large stomach of B. gemesi. Short stature homobox protein, or SHOX, and high mobility group protein, HMG, genes in this pathway were expanded. A lack of these causes dwarfism, therefore an increase in these should cause giganticism. Enriched genes related to nutrient storage, absorption, and utilization were found in the digestive system of the giant isopod. These genes are important for insulin signaling and resistance, endocytosis, and glycolysis. This would mean they adopted an efficient mechanism of glycolysis to provide sufficient energies for activities of such a large animal without a large food source. Endocytosis is an important process of vesicular transport which aids in the absorption and utilization of food. The researchers also found genes related to lipid degradation were significantly decreased, meaning B. gemesi accumulates lipids due to the low efficiency of degradation. So what conclusions can be drawn from this study? B. gemesi's genome shows the variable adaptations of deep-sea organisms. These organisms undergo selective pressure, which could have shaped their genome. Their environment or body size could explain their genome size. It could also be explained by low basal metabolic rate, low mobility, and high longevity of B. gemesi. The factors that drove this genome size to be so large are complex, and not one alone can explain it. B. gemesi achieved a balance of low BMR, specialized energy storage organs, and efficient nutrient absorption and utilization. These have helped this giant isopod to not only survive, but thrive in the deep sea. Thank you for watching, and see you next time as we uncover more deep sea mysteries.